Welcome back, everyone, to the Immunology for Everyone podcast by SKA. Transcending ahead into the world of immunoglobulins and antibodies, your host and subject matter expert, SKA, today sheds light on how the immune system memorizes details of these infectious agents. Let us understand how it recognizes the foreign agents when they strike again and again, and what follows once the cascade begins. Do not forget to get your peers into listening, and do not forget to share your views and queries. Over to SKA now.
Dear friends, when the SARS-CoV-2 appeared a few years back and started infecting people, we all keenly observed that the antibodies against SARS-CoV-2 in these individuals showed weak neutralizing capacity and also waned off very quickly. So within 3 months, the antibody titers were undetectable. But it was good to find that the antibodies sustained for much longer time in individuals who got the vaccine shots or those who got a second exposure. So the researchers who tried to find out the details of these antibodies, they even showed that the neutralizing capacity of the antibodies in such individuals was also improved manifold after vaccination or in the individuals who had hybrid immunity, like those who were initially vaccinated and got a super infection later on, or those who had in fact got a cycle of infection and got vaccine later shots later on. So let's try to understand what are the mechanisms behind this change in stance of the antibodies produced against the same virus in different situations. In this context, the antibodies being the effector molecules released by the B cells, in the last episode we discussed the phenomena of linked recognition for B cell activation for those antigens which are called the thymus dependent antigens, and there is a mandatory requirement for T cell help. And we find that the activation of B cells in this manner leads to clonal expansion of antigen specific B cells with the formation of primary focus of antibody forming plasma cells. So now we discuss the interesting phenomena called the affinity maturation, and this will help us understand how this leads to generation of antibodies with increased binding and neutralizing capabilities that is seen after vaccination or repeated infection in SARS-CoV-2, or for that matter any other infection. It has been shown that after interaction with antigen specific T cells, the proliferating B cells, they differentiate into antibody making plasma cells and they migrate to the red pulp of the spleen or medullary cords of the lymph node. The differentiation of B cells into a plasma cell in this manner is accompanied by many morphological changes that reflect the commitment to the production of large amounts of secreted antibody. So the plasma cells by nature are rich in cellular machinery to make large amounts of protein in the form of secreted antibodies, but these cells can no longer present the antigen to the helper T cells now. However, the surface immunoglobulin is still expressed on these cells though at a very low level, and recent studies have suggested that the survival of plasma cells may be determined in part by their ability to continue to bind to the antigen, and this surface immunoglobulin molecule helps in that process. So interestingly, these plasma cells have a range of lifespan. While some of the plasma cells they survive only days to few weeks after their final differentiation, and also so-called these are the cells which are called the short-lived plasma cells, whereas there are other cells which are very long-lived plasma cells and account for the presence of persistence of antibody responses for longer periods of time. So, understanding the fate of B and T cells that proliferate in the primary focus, some of these cells they migrate into a primary lymphoid follicle where they continue to proliferate and ultimately they form a germinal center. For your information, the germinal center is a sort of an island of cell division that sets up among the large number of resting B cells in the primary follicles. These proliferating B cells, they displace the resting B cells which are not dividing towards the periphery of the follicle, forming a sort of mantle zone of resting cells around the germinal center. A germinal center is mainly composed of proliferating B cells, but antigen-specific T cells, excuse me, also make up about 10% of the lymphocytes in the germinal center, and they provide indispensable help to the B cells for their activation. So besides the resting B cells, the primary follicles also contain a dense network of other cells like follicular dendritic cells, which are also called the FDC, which play a very important role in maintenance of these follicles. So we have the B cells which are proliferating, some T cells and follicular dendritic cells. So as these follicular dendritic cells, they attract the naive and activated B cells into the follicle by secreting a chemokine which is called the B cell attracting chemokine, or also called the CXCL13. So these early events in the primary focus lead to the prompt secretion of specific antibody that serves as the immediate protection in infected individuals. And that's what we discussed about the SARS-CoV-2 initial infection, where the antibodies they appeared, but they waned off quickly. But those are the antibodies which were made by these kind of plasma cells. However, the germinal center reaction, which involves the induction of T follicular helper cells which express BCL6 transcription factor, which in turn help the induction of BCL6 expressing B cells in the germinal center and provides for a more effective response at a later stage. If the pathogen establishes a chronic infection or the host becomes reinfected, in those conditions, these germinal center reactions they occur and form a specific type of these B cells which are proliferating there. And in that situation, the B cell undergoes a number of important modifications in the germinal center, and these include the somatic hypermutation, affinity maturation, and isotype switching. So let's understand each one of these, how it occurs. The affinity maturation is the process by which the B cells, which have been activated by the T follicular helper cell, produce antibodies with the increased affinity for the antigen during the course of an immune response. So with repeated exposures to the same antigen, the host will produce antibodies of successively greater affinities. So every time a secondary response can elicit antibodies with several fold greater affinity than the primary response. So, the affinity maturation primarily occurs on membrane immunoglobulin B cells in the germinal center. So, the process of affinity maturation is thought to involve two interesting interrelated processes occurring in the germinal center of the secondary lymphoid organs. The first one is the somatic hypermutation. This phenomena include the mutations in the variable antigen binding sequences, which we earlier described as the complementarity determining regions, or the CDRs. And I told you that they are the ones which bind to the antigen, and they are present on the variable region of the immunoglobulin molecule. So this is called hypermutation because the mutation rate here is up to 1 million fold higher than the cell lines which are present outside the lymphoid system. And although the exact mechanism for somatic hypermutation is not yet completely known, a major role for the enzyme like activation induced cytidine deaminase has been described to play some important role. So the increased mutation rate results in 1 to 2 mutations per CDR, and hence per generation of the antibodies. So there's a lot of mutation which occurs, and it makes the CDRs genetically changed, and these mutations alter the binding specificity as well as the binding affinities of the resultant antibodies. And the second important phenomena which occurs is the clonal selection, which means that the B cells that have undergone somatic hypermutation must compete for limiting growth resources, including the availability of antigen and signals from the T follicular helper cells. The follicular dendritic cells, on the other hand, of the germinal center, they present the antigen to the BCL6 positive B cells, which undergo the clonal expansion. So follicular dendritic cells are incidentally an important subset of stromal cells that are located within the B cell follicles and germinal centers of secondary lymphoid organs. And these are actually distinct from the bone marrow derived classical dendritic cells, because these cells are a non-phagocytic type and they don't migrate as the classical dendritic cell they do. And these have several different functions, including activation of B cells and maintenance of immunological memory. So unlike other dendritic cells, these follicular dendritic cells, they do not process antigen for presentation to the T cells. Rather, they stimulate the CD4 positive Th2 type of response and maintain memory by a unique mechanism. Using a repertoire of receptors, these follicular dendritic cells bind the processed and unprocessed antigens onto the beaded three-dimensional structures, which are also called the immune complex coated bodies or icosomes. So remember that icosomes are present on the follicular dendritic cells, which contain the antigen, and they basically serve as the depot of antigen for continuous supply to the B cells for activation and release of antibodies. So interestingly, the antigen may be retained in icosomes for many months or perhaps years. Then comes the positive selection. So the daughter cells with the higher affinities for antigen, having gained a competitive advantage, are favored for positive selection, and that leads to their survival. So positive selection is based on steady crosstalk between the T cells and the germinal center B cells, which present the antigen on its surface. So germinal center B cells, they present the antigen, display that, and T follicular helper cells, they recognize it and interact with that. Because we have a very limited number of T follicular helper cells inside a germinal center, only highly competitive B cells stably conjugate with the T follicular helper cells excuse me, and receive T cell dependent survival signals. The daughter B cells, on the other hand, that have undergone somatic hypermutation but bind the antigen with a lower affinity, are outcompeted and they are deleted. Over several rounds of such selection, the resultant secreted antibodies have effectively increased affinities for the antigen. So there are many, evidences for positive and negative selection in the pattern of somatic hypermutations in variable region of B cells that have survived passage through the germinal center. If we talk about the negative selection, it is shown that loss of cells that had mutated to cause the misfolding of the immunoglobulin variable region, and that way turn out to be low affinity receptors. So the negative selection is an important force in the germinal center, which eliminates about half of the cells which are basically expressing very low affinity receptors. Because if there was no negative selection, B cells which are dividing 3 to 4 times every day in a single germinal center would quickly create enough number of cells to overwhelm the whole system, as it will lead to create a more than 1 billion cells in about 10 days in a single germinal center. So, but instead we know that the germinal centers actually contain only a few thousand B cells, even at its peak. And on the other hand, the mark of positive selection is an accumulation of numerous amino acid replacements in the complementarity determining regions which bind to the antigen. So the consequence of these cycles of proliferation, mutation, and selection, all happening within the germinal center, is that the average affinity of a population of responding B cells for its antigen increases over time, which explains the phenomena of affinity maturation of the antibody response. So the selection process can be quite stringent, because although 50 to 100 B cells may enter a germinal center initially, but most of these cells die and leave no daughter cells. And by the time the germinal center reaches maximum size, it is typically composed of the descendants of only one or maybe a few antigen-specific B cells. So, what we finally understand about the germinal center B cells is that these cells are inherently prone to die. And in order to survive, they must keep receiving specific antigen signals. And it was originally discovered by in vitro experiments that the germinal center B cells could be kept alive by simultaneously cross-linking of their B cell receptors and ligating their cell surface molecule, the CD40. And under in vivo conditions, the cross-linking of B cell receptor could be done by the antigen. And the additional signals of CD40, CD40L interaction through direct contact with the T follicular helper cells. So this implies that continuous antigen delivery is important for the sustainment of germinal cells. So, and the source of antigen in the germinal center has been a really matter of some controversy. Here it has been assumed that the antigen which is shown to be trapped and stored on the surface of follicular dendritic cells in the form of icosomes is helping to sustain the germinal center B cell proliferation. But this may be true under only certain circumstances, because there are few studies which have shown that the antigen on follicular dendritic cells may not be required to sustain a normal germinal center response. In fact, the role of antigen depot on these cells is not well understood yet, although it could be important to maintain long-lived plasma cells under some circumstances. So the question still remains is that where does the antigen that sustains the germinal center B cells come from? From where does it come from? Under normal circumstances, it must be most likely that the live pathogens carried to the lymphoid tissue and multiplying there will continue to provide the antigen until they are eliminated by the immune response, after which the germinal centers they decay off. And as a supportive evidence, we know that the vaccination with protein antigens are usually given in a formulation that makes a depot and slowly release the antigen over time, which mimics the situation with live pathogens. In fact, it is difficult to stimulate germinal center formation by immunization when we are giving a vaccine without either a live replicating pathogen or a sustained release of antigen which is given along with an adjuvant. So that's why we use the adjuvant when we are giving a vaccine, and adjuvant plays a very important role for doing all this, depot effect and all. But how these signals that maintain the germinal center and exert their effects on the B cells is not yet completely understood. So although it has been shown that the combined signals from the B cells receptor and the CD40 seem to upregulate a protein which is the BCL6 or BCLXL, which promotes the survival of B cells in the germinal center. So there must be, however, many other signals that may be promoting B cell differentiation, which remain to be discovered. So now we understand that BCL6 expressing T follicular helper cells are absolutely required in maintenance of germinal center for proliferation of long-lived plasma cells. So here, let me quote one of the very recent studies, and that is again about the SARS-CoV-2 infection, which will really explain the importance of BCL6 positive T follicular helper cells in maintenance of germinal centers for proliferation of long-lived plasma cells. So in general, we understand that some pathogens, as we can call them the immunogens also, will induce a B cell T cell interaction leading to differentiation of BCL6 T follicular helper cells, which move into the germinal center and keep interacting with the B cell and follicular dendritic cells for proliferation of mature plasma cells in the germinal center. But during acute SARS-CoV-2 infection, the virus induces the differentiation of inflammatory Th1 cells that make large amount of inflammatory cytokines, including TNF alpha, in the vicinity of the B cell T and B cell interaction in the lymphoid organs. So the TNF alpha basically inhibits the differentiation of BCL6 expressing T follicular helper cells that leads to disruption of germinal center, leading to stunted interaction of T follicular helper cells with the plasma cells and further causing no proliferation of long-lived plasma cells. So in these conditions, the long-lived plasma cells are not surviving. And you have only so whatever antibody response we see in COVID-19 in the early stages initially was because of the antibodies made by the short-lived extrafollicular plasma cells, and that basically wanes off quickly as the long-lived plasma cells are not proliferating due to the disrupted germinal centers. The COVID-19 vaccines, on the other hand, would induce formation of mature germinal centers with a sustained antibody response from proliferating long-lived plasma cells in the germinal center. So that basically answers the questions we started with, that why the antibodies response is short-lived in initial infection but improves manifold after vaccination. Now, what about the memory B cells? Some surviving germinal center B cells, they differentiate into memory cells, and these cells, they divide very slowly and express surface immunoglobulin with specificity to same antigen, but they do not secrete the antibody at a high rate because they become the dormant population of cells which carry the memory for the antigen. And these memory B cells inherit the genetic changes that had occurred in the germinal center cells, including the somatic hypermutation as well as the gene rearrangements that result in the isotype switching. So the signals that control which differentiation path a B cell will take is not very well understood yet, but there are a few indications in the literature. One is that in this context, it has been proposed that the signals from follicular dendritic cells are important in stimulating a B cell to become a memory cell. Although it has been shown that the memory cells can still develop, maybe at a very lower frequency in a mouse which is mutant, which lacks the follicular dendritic cells. And this indicates that there may be other mechanisms also involved in this process. Another speculative possibility is that the affinity for antigen controls the B cell differentiation. And it basically indicates that with high affinity cells perhaps being preferentially stimulated to become the memory cells, while the lower affinity cells are allowed to undergo further cycles of proliferation, mutation, and then selection as a high affinity cells. So this is also remains as one of the mysteries of germinal center that immunologists have yet to resolve. So guys, let's summarize what we have discussed about thymus dependent antigens and B cell activation. For activation of B cells for thymus dependent antigens, the initial interaction occurs in the T cell area of secondary lymphoid tissue, where both antigen specific T helper cells and antigen specific B cells are trapped as a consequence of binding the same antigen, and the phenomena is known as linked recognition. Here the T helper cell differentiate into BCL6 expressing T follicular helper cells. Further interaction with this T follicular helper cells and B cells, they occur after migration into the B cell zone or the follicle and formation of germinal center. So the T follicular helper cells induce a phase of vigorous B cell proliferation and direct the differentiation of clonally expanded progeny of the naive B cell into either antibody secreting plasma cells or memory B cells. So during differentiation of activated B cells, the antibody isotype can change in response to cytokines released by the T follicular helper cells, and the antigen binding properties of the antibody can change by somatic hypermutation of the variable region genes. Further, the somatic hypermutation and the selection of high affinity binding occur in the germinal centers. So the interaction with BCL6 positive T follicular cells is absolutely important because these cells, they control the processes by selectively inducing BCL6 expression in the B cells that have retained their specificity for the antigen by inducing proliferation and differentiation into long-lived plasma cells and memory cells. So guys, we close here today, and we'll discuss the B cell activation for T independent antigens in the next episode. Till then, stay tuned, keep listening, and thank you very much. Goodbye.